Today we're going to be talking about Punnett squares and the monohybrid cross. Review from previous lectures include meiosis and sexual reproduction. We talked about Gregor Mendel and his laws of inheritance, hybridization, whether or not a allele is heterozygous or a homozygous dominant, dominant and recessive alleles and traits, and the basic probability of inheriting a trait. Today we're going to be talking about how we access an organism's phenotype and their genotype. A phenotype is an observable trait of an organism. Examples would be a hair color, freckles, or dimples, as we can see in the difference in hair colors to your right with the dog's hair color. Genotypes refers to the genetic makeup or combination of alleles inherited from parental organisms, aka the letters. So you can see on your right, just below the picture of the puppies, a capital B and lowercase b. Capital B would represent a dominant allele, and a lowercase b would represent a recessive allele. But what are Punnett squares? Punnett squares are graphical representations that are utilized to identify the probability of an offspring inheriting a specific trait from the combination of parental organisms where their genotypes are known. For example, if we're looking at the Punnett square to your right, we're looking at the color of pea pods. A capital G shown in green would indicate that that pea pod is going to be green. And if a capital G is expressed, then anything in the genotype that has a capital G is going to be a green pea pod. A lowercase g would indicate that it's going to be a yellow pea pod. And if we're looking, you can see that you have to have at least 2 yellow G's for the phenotype, what is shown to be yellow. If a capital G is present, then the pea pod will be green. So what is a monohybrid cross? A monohybrid cross is a cross between two organisms, two parents, in which one characteristic trait of an organism is in question. For example, we're looking at eye color, hair color, freckles, and or no freckles. So we're only questioning one thing. So for example, if we're looking over here at the Punnett square on your right, we have eye color in question, whether or not the organism is going to get brown eyes or blue eyes. Brown eyes being dominant, and if the capital B is present, then you will get brown eyes. Blue eyes being recessive, and if the lowercase b is present twice, then you will get a blue eye. Let's look at a demonstration. A cross between a homozygous dominant purple flower, meaning 2 capital letters, and a homozygous recessive white flower has occurred. Homozygous recessive meaning 2 lowercase letters. What is the genotype ratio of the offspring? The phenotype ratio. So we're going to look at an example real quick of how to set up your Punnett square. The first thing you're going to do is make a box and divide it into 4 squares. We'll take the homozygous dominant genotype, meaning our letters, and we're going to represent those as being B's. So it is homozygous dominant, so 2 capital B's, mixed with a cross between a homozygous recessive, 2 lowercase b's. And we want to know the genotype ratio of the offspring and the phenotype ratio. So the first thing you do is bring down a letter from the top. In the first box, you'll get a capital B, and one from the side. You get a lowercase b. You bring one down from the top for the second box, and one from the side. So our first 2 boxes are big B, little b, big B, little b. Then for our third box, one from the top again, so capital B, and lowercase b. And then one from the top and one from the side again, you get capital B and lowercase b. So our Punnett square is done. We've dropped all of our letters in their appropriate squares. And these squares represent the likelihood that that is the probability that they're going to get this trait for color of the flower. Capital B meaning that if it's present, it's going to be a purple flower. And you have to have at least 2 lowercase b's for it to be a white flower. So in this situation, our genotype ratio is going to be heterozygous. So all of these are going to be heterozygous because you have a capital B and a lowercase b for all of them. So your ratio would be 4 out of 4. Then if we're looking at the phenotype ratio is going to be all purple flowers because a big B is present in all of our boxes. So the phenotype ratio would also be 4 out of 4 for purple flowers. So in future lessons, we're going to be talking about how to perform a dihybrid cross, where we're talking about more than one trait. So for example, the pea pod shape and whether or not it's wrinkled and or smooth. Alternatives to dominance and recessiveness. What happens when a trait is not completely dominant? We'll talk about multiple alleles, X-linked disorders, sex-linked disorders, and we'll talk about pedigree charts and what happens when inheritance goes from generation to generation. And we'll see you in the next lesson.